So Veri started her career in the pharmaceutical industry, specializing in quality assurance for GMP drug manufacturing. She later moved on to strategic roles in the healthcare industry. Currently, she is managing the product portfolio of nucleic acid chemistry in LGC Biosearch Technologies. Veri holds both a Master and Bachelor in Science in Biomedical Sciences from Bern and Freiburg University, respectively, and also did PhD studies in Medical Neuroscience from the Charité University Hospital. Moving on to Shireen. Shireen started with LGC Biosearch Technologies as a chemist in 2016. Throughout her years being a chemist, her role has been primarily lab-based as part of the production team, working on the synthesis of LGC's catalog products at both Belshore as well as Petaluma sites in UK and US respectively. She's currently the product manager for nucleic acid chemistry portfolio. Shireen graduated from University of Strathclyde, Glasgow, with Bachelor of Science in Chemistry, specializing on drug discovery. So Veri and Shireen, over to you. Let us know if you can hear us and your audio is loud and clear.
Thank you very much, Joy, for this nice introduction. I hope you can hear me well. Can you hear me?
Yes, loud and clear.
Okay,
The stage is all yours,
excellent.
Veri.
Excellent. So good morning, everyone. I hope, apologies if I don't look too fresh. It's actually almost 4 a.m. in the morning for me. But I'm very glad that I was invited to this event, and hopefully I can give you some useful insight into oligonucleotide-based therapeutics. So for the ones who are maybe less familiar with these technologies, I'd like to first start with a little introduction about oligonucleotide-based therapeutics. So these comprise various classes of drugs.
Veri, sorry to interrupt. Just give me a second. I'm trying to get you onto the gallery top. Just give me a second. Let me stop sharing and then reshare again. All right, here you go.
All right, if you could actually already move to the next slide, that would be nice. Perfect, thanks. So oligonucleotide therapeutic comprise various classes of drugs that are made of chemically synthesized oligonucleotides. And these compounds can modulate gene expression as well as impede protein function by diverse mechanisms. So among them, 18 sense oligonucleotide as well as siRNA oligonucleotide are the one which are were developed the furthest, and those are the one I'm going to talk more about in this presentation. So next slide, please. Next slide, Joy, please. Thank you. So siRNA stands for small interfering RNA. They are usually between 19 to 25 nucleotides long and are usually double-stranded. And they are comprised of a guide strand, also called the sense strand, as well as the passenger strand, which is the passenger strand, which is the sense strand, and the guide strand, which is the antisense strand. And how they work is that once they're introduced into the cellular space, usually by means of a delivery system, such as the nanocarriers, they are then loaded into the RISC, which stands for RNA induced silencing complex, and once in the RISC kind of unwind these double strands and remove the passenger strand, which is then degraded, and the guide strand is then used to help the RISC find the target mRNA. And then activate RNAs to degrade the targeted sequence. Sorry. And whereas ASOs stands for antisense oligonucleotide, they are usually or they are all the time single-stranded and can be from 12 to 25 nucleotides long. However, shorter than 12 nucleotides can trigger off-target effect, and longer than 25 nucleotides is also usually not used, as it reduces intracellular uptake of those compounds. So ASOs can work via a virus type of mechanism. 3 of them are kind of the main ones. So one is by activating RNase H that can then degrade the target mRNA. Another way is for the ASO oligo to firmly bind to the target mRNA, and then it prevents the attachment of the ribosome complex, so there is no translation into proteins. And then one other way of regulating the gene expression is by actually working on the pre-mRNA and regulating the splicing. Next slide, please. So although all this looks pretty easy in theory, in order for oligonucleotide-based therapeutics to actually become successful, a lot of challenges need to be overcome. So the problem is that with unmodified or naked nucleic acid, they are not surviving very long in the body. So one reason for that is that they are immediately degraded by various endo and exonucleases, and they are cleared via renal filtration, and they can also induce an immune response, and it's also, depending on the target organ, it's difficult for the oligonucleotide to penetrate the different tissues, especially if the endothelial membrane is very tight, like in the blood-brain barrier. Then one other challenge is actually the uptake into the cells, which most of the time happens via endocytosis. But then, once again, the oligonucleotide has to find a way to escape the endosome before it gets degraded. So all those are challenges which need to be overcome. Next slide, please. So despite all these challenges, as of today, already 14 oligonucleotide-based therapeutics have been approved by either the FDA or the EMA, and many more are in clinical development. And what is great is that they can actually target many diseases in various indications. So not only, like Joy mentioned, not only rare diseases, but now it has been proven that it can also target chronic diseases. Next slide, please. So this is just an overview of all these 14 compounds that were so far approved. And as you can see, most of them are made of sRNA or ASO oligonucleotide. But I won't go further in details. So next slide, please. So what does it take to actually design such oligonucleotide? So first of all, of course, you need to find, to design the sequence, which is complementary to the target sequence of the gene that you want to target. But then this is not sufficient. Like I mentioned, if you just leave it as it is, your oligonucleotide won't survive long in the bloodstream or inside the body. So you need to add specific chemical modification to improve the stability of your oligonucleotide and its half-life. So for that, there are different modifications that can be used, and I will tell you more about it. And another way is to make sure that your oligonucleotide is actually delivered to the target organs and cells. So for that, most of the time, you need to find the proper delivery system that will actually help you achieve that. Next slide, please. So let's start with the designing antisense oligonucleotide. So since this mechanism was discovered a way back, different modifications have been developed over time, and they can be categorized under 3 different generations. So next slide, please. Next slide, please. Thank you. So the first generation is actually about replacing phosphodiester linkage by other type of linkages. So, for example, it can be replaced by methylphosphonate as well as phosphoramidate and phosphothiorate linkages. And all of them actually increase the stability toward digestion by nucleases of the oligonucleotide. However, they are all a bit different in their features. So, for example, methylphosphonate and phosphoramidate are both making the backbone neutral, whereas phosphodiester are usually negatively charged. So this further improves intracellular transport as well as resistance to nucleus degradation. However, the disadvantage of those 2 modifications is that it interferes with the activation of the RNase H, which is needed to degrade the targeted mRNA. So the phosphorithioate are actually the modification of the first generation, which is the most widely used, and it has 2 roles. So the first one is to increase resistance to nucleus digestion, and the other one is to increase binding to different proteins in the plasma and the serum. So, for example, albumin and heparin, and by doing so, it actually increases the half-life in the body. So the problem also with phosphorate linkage is that they are pretty toxic. However, the big advantage is that it's one of the only modification that support RNase H activation. And fomibersen, which was actually approved in 1998 as the first ASO to be approved, actually has its structure fully modified with phosphothiorate linkages. So like I said, these phosphorate linkages are very needed in order to increase the resistance to nucleus degradation. However, they are pretty toxic. So then researchers decided to further improve the chemical modifications, and that's why they design new modification that falls under the 2nd generation. So next slide, please. So for the members of the 2nd generation, they are characterized by the replacement of the hydroxyl group at the 2' position of the ribose with methyl or methoxyethyl groups, which leads to the formation of 2' O-methyl or 2' methoxyethyl nucleotides. And so this modification actually makes the oligonucleotide less toxic. It also further increased the resistance to nucleus degradation, and it increases the affinity of the oligonucleotide to its target RNA. And a way to even further increase resistance to nucleus digestion is to actually combine those modification of the second generation with phosphorothioate linkages. So this was the case for NISD resin. However, the problem is that when the oligonucleotide is fully modified with this ribose modification, then it can no longer activate the RNA H enzymes to degrade the mRNA. So a way to remedy to this is actually to leave the middle section of the oligonucleotide without ribose modification. So it's just unmodify deoxyribonucleotide, which can then activate the RNase H enzyme. And then on the side, to improve the stability, you have the ribose modification. So this is called, the gapmer. And although modification of nucleobases are not so common in antisense oligonucleotide, for example, you can still exchange the cytosine with a methyl cytosine so that it increases the stability of the oligonucleotide and increase the affinity to its target mRNA. Next slide, please. So in the third generation, further modification were discovered. So for example, there's the log nucleic acid in which the 2' oxygen and the 4' carbon on the same ribose actually are connected through a methylene bridge. And this modification improves the stability of the oligonucleotide through increased melting temperatures. So similarly to the second generation modification, when an oligonucleotide is fully modified with LNA, it can no longer activate the RNase H enzyme. So for that, it's also useful to create a gapmer with having the LNA modification just on the wings of the oligonucleotide and leaving the central region just with DNA. Other modification in the third generation comprise peptide nucleic acid as well as phosphoramidate morpholino oligonucleotide. And these two are neutrally charged molecules, and they do not show high levels of plasma protein binding and are rapidly cleared by renal filtration. So this is not really an advantage. However, they can increase the melting temperature, and by such the affinity of the target RNA for the target RNA is improved. So for example, eteplirsen, which is an approved drug, is based on the morpholino technology, and it's used for the treatment of Duchenne muscular dystrophy. Yes, let's move to the sRNA design. So initially, studies showed that fewer modifications were required to design a potent sRNA. However, over time, more and more chemical modifications have been added to the sRNA, mostly 2' O-methyl and 2' fluoro, as well as phosphorothioate linkages. So next slide, please. So similarly to antisense oligonucleotide, phosphorothioate linkages are very important and used a lot to design siRNA. That's because, like in ASOs, they can increase the stability of the oligonucleotides towards RNA degradation. However, they do aggravate chemical toxicity. So for that reason, it's very important to not overuse them and to place them in targeted location. So the initial design was to just include only 2 of those linkages on the antisense strand. However, if we take this example picture here, so we can take a look at revusiran as well as givosiran, which have a pretty similar chemical composition. However, revusiran was removed from clinical trial after it appeared that there were more deaths in the treated groups as in the placebo group. Whereas givosiran was recently approved as a drug. So if you have a look at the differences between these two molecules, you can see that first of all, givosiran has 4 more phosphorothioate linkages, which were added at the 5' end of the sense and antisense strands. And another difference is that givosiran has fewer 2'F modification, which actually increased the toxicity of the oligonucleotides. So it's better not to have too many of those. And so these small changes actually allowed to, first of all, to have a drug which is now approved and used to treat patients. And moreover, it requires lower dosage and at lower frequency, so which is a big advantage. So next slide, please. So one other difference between these two drugs or compounds is the addition of 5' vinyl phosphonate on givosiran. And the reason for that is because the 5 phosphonate is usually removed by phosphatase, and this impairs the stability of the oligonucleotide and impairs the loading of the siRNA into the RISC. So by adding this mimic of 5 and phosphonate, it can improve the silencing activity of the oligonucleotide. Next slide, please. So other modifications that are also very important, like I mentioned earlier, in designing siRNA are 2' ometyl and 2' fluoro modification, which are ribose or modification of the 2' hydroxyl group on the sugar. So the only thing to note is that if you have a guide strand which is fully modified with 2' ometyl modification, the strand is then completely inactive. As well as if it's fully modified with 2' fluoro modification, it has a really reduced activity. So you don't want to do that. Instead, the best is to alternate those 2 modifications within the oligonucleotide. And then, like I mentioned earlier, even better is to have a higher amount of 2' ometyl modification versus 2' fluoro modification. So next slide, please. So a good pattern example is the standard template chemistry, which was designed and established by Anilam. So this was the first version, and then they improved it. So as I mentioned, the first one had only 2 phosphorothioate linkages, and then the improved version added 4 more linkages, and then the advanced version removed the number of 2' fluoro modifications, and then the latest version even adds a GNA modification, which is a glycol nucleic acid modification, which destabilizes the oligonucleotide thermally and this inhibits the induced hepatotoxicity. And then additionally to those modifications, there are many more which are now being researched, and which are for some of them which are being studied in different clinical trials, so such as unlock nucleic acid as well as lock nucleic acid, which confers various properties to the siRNA. Next slide, please. So Shireen already told you quite a lot about the different delivery systems that can be used to enhance and target the organs that you want your oligonucleotide to be able to reach. So, like she mentioned, lipid nanoparticles have been used a lot so far, as well as conjugating different lipid to your oligonucleotides, such as cholesterol, tocopherol, palmitate, and so on. You can also use aptamer conjugates, as well as antibody conjugates, peptide conjugates, and some other even more original modifications such as DNA cage, spherical nucleic acid, and exosome, which can carry the oligonucleotides and bring them into the intercellular space. And of course, GalNAc, which is the flagship for liver delivery. So with that, I would like to thank you all for listening, and I'm happy to answer any questions.
Hi everyone. Thank you for having me. I'm Shireen. I'm a nucleic acid chemistry product manager at LGC Biotech Technologies. And building on from what Valerie was delivering, I'll give you some information around the GalNAc modification, followed by solid support choice for oligosynthesis, and then finally ending with some current work and advancements that are taking place within the therapeutic field. So to start, I'm sure most of you have heard of GalNAc. As it stands, it's the only means by which we can target a specific organ within the body for delivering oligos, that organ being the liver. So Valerie touched on this, but some reasons why RNA-based therapeutics struggle to cross the cellular membranes is that they're fairly large in size and also quite anionic, in addition to being unstable and also lacking in resistance to nuclease degradation. So to combat these issues, research and development has led us to GalNAc, which is an amino sugar derivative that allows siRNAs to be delivered to the liver. It does this by specifically binding to ASGPR, which is a cell surface membrane glycoprotein found in liver cells. So once a pH drop is triggered within the cell, ASGPR releases the GalNAc siRNA conjugate and then rises back into the surface of the cell to be reused again. This ability of the ASGPR receptors to be recycled is what's thought to be contributing to the efficient delivery of GalNAc. So next slide, please, Joy. So we as LGC Biotech Technologies offer two GalNAc products within our catalog, one being a phosphoramidite and the other is a CpG solid support with the option of two pore sizes. So these products were developed in partnership with Mina Therapeutics and also LGC Axo Labs. The products were designed and made at our Bells Hill site in Scotland, and then the oligoconjugates were prepared and optimized by Axo Labs and finally tested by Mina Therapeutics. So the key features of our GalNAc monomers is that they can be used to build up flexible GalNAc clusters with varying numbers and position of the monomers within the oligo. They have a deoxyribose feature, which fits directly into the natural sugar phosphate backbone with minimal disruption to the duplex. So as I mentioned before, the CPG product is offered in two different pore sizes, depending on what the end use of the final oligo is, which I'll touch on briefly in our next topic as well. But they also feature a succinyl linker, which makes them quite easy to cleave by using concentrated ammonium hydroxide at room temperature. So next slide, please, Joy. Okay, moving on. I'd like to now talk about the solid supports used in oligosynthesis. So these are generally split into either polystyrene or controlled pore glass, depending on the functionality of the oligo. Usually antisense oligos, which are short and simple in design, are made on polystyrene, while sRNA are primarily synthesized on CPG. So on the right here, we just have a table of drugs in various clinical stages where the solid support of choice has been primarily CPG, treating a variety of diseases from acute hepatic porphyria to viral infections like hepatitis B. It's also worth noting that the vutrisiran was very recently approved by the FDA under the commercial name Amvuttra. So next slide, please, Joy. Okay, so in this slide, I'd just like to talk about how to pick the right pore size of your CPG solid support, which we have available in a variety of pore sizes and nucleosidic loadings at LGC Biosearch Technologies. So the pore size of choice is usually dependent on the oligo design itself. If you're building shorter oligos, for example, less than 30 bases long and looking for high yields, you're best using 500 angstrom. Whereas if you're building longer oligos, you'll require larger pore sizes. So from that, when it comes to specific uses, 500 angstrom to 600 angstrom is very well suited for therapeutic oligos, whereas higher pore sizes are best used for oligos that are built for diagnostic and research purposes. Next slide, please, Joy. And as a final bit on solid supports, I'd like to just give a quick mention to universal supports, which are supports where there is no nucleobase or modification retained at the 3' end of the oligo sequence after cleavage. So this allows for many advantages, such as the ability to be utilized in any oligo sequence, since the first base is determined by the first phosphoramidite addition in the cycle. And then when preparing wells in plate synthesizers, this prevents the placement of the incorrect resin in a well, which allows for automated preparation of the plate to stock, just ready for your synthesis. It's also quite beneficial when it comes to large-scale synthesis, as it allows for a simplified supply chain, since you only have one solid support and you don't need to keep a stock of all bases. So at LGC Biosearch Technologies, we offer a variety of universal supports. However, our most effective product is definitely our isopropyl version, which is similar to a phenyl product that does go by the name Unilinker, but has some advantages, which is that the removal of the linker from the 3' terminus is 5 times faster than that of the Unilinker product. There are also advantages to the unprotected solid supports versus the dimethoxytrityl protected supports. For example, the lack of linker cleaved during the first detritylation step with dimethoxytrityl protected groups. And also the DMT group in universal supports is so stable that the initial detritylation step isn't quite necessary, although this means that users have to create a custom protocol for the detritylation step. Next slide, please, Joy. Okay, so moving on from GalNAc and CPGs, I'll now take you through some current and future developments that are ongoing within the therapeutic oligo space. So starting with lipid nanoparticles, which are currently being used for many applications, for example, protein replacement therapy and also many vaccines under development at the moment. So these developments in current work has been further enhanced, certainly by the success of Onpattro, which is the first RNAi drug. The examples of vaccines being developed aim to treat many known diseases such as influenza, Zika virus, malaria, but also coronavirus, which we touched on at the very start. We're also seeing development within personalized vaccines for cancer. Some are already in early clinical stages to treat both breast cancer and melanoma. Next slide, please, Joy. It is also worth noting that LNP vector technologies, along with AAV, are currently being utilized for gene editing as well. To treat, for example, retinal dystrophies, HATTR, and metabolic disorders such as phenylketonuria, which is showing promising results in mice. Next slide, please, Joy. And in this slide, I'd just like to highlight a number of diseases that are currently being targeted by treatments utilizing antisense oligos. So, in addition to rare technologies, rare neurological disorders, antisense oligos are also being utilized for viral infections like hepatitis B and coronavirus, but also diseases such as cystic fibrosis, hemophilia A, and so on. So, next and I think final slide. Yep, thank you. So just to wrap up, finally, I'd just like to just mention that although oligotherapeutics are showing remarkable results and high potential in treating rare and prevalent diseases, there's still a number of issues that we need to try and resolve within this field. The main one being that, as it stands, we can only specifically deliver oligos within the body, and that's the liver through GalNAc, which I've mentioned earlier. Although it's worth mentioning that this is certainly a hot topic, and a lot of research is being conducted both academically and within the industry to find the quote unquote next GalNAc. In addition, there's still room for improvement within toxicity work/studies. And finally, we only have a limited set of clinical data for many of these treatments, since testing is only carried out on a small number of patients rather than large amounts, due to majority of these treatments being for rare diseases. Although, again, that is starting to change now as well. So that's all from me. Thank you for listening, everyone, and let me know if you have any questions.